Hello, this is part 2 of chapter 24. We're going to begin looking at the small intestine and large intestine. So the normal flora of the small intestine is going to exist, but not as much and as plentiful as the large intestine. The small intestine, the pH is neutral, so it does harbor some normal flora, not as many as the colon as I mentioned. And the small intestine is really designed to do all the digesting and the absorbing of the nutrients that we eat. So the surface of the small intestine looks like this. It's got tiny finger-like projections called villi, and the villi exist to increase surface area so that the nutrients from the food can be absorbed all along the villi, right? So it can be absorbed from the sides, from the top. So every sort of mucous membrane that the food can touch can serve to absorb the food. So we're really, really good at, maximizing absorption of the calories and nutrients from our food. Because it does receive foods from the stomach does have an acidic pH. So the small intestine has to neutralize that pH. And so that way the acids don't burn the lining of the small intestine. So the pancreas actually helps to neutralize the substances. Yeah, so basically the small intestine is neutral. It is made to absorb nutrients, absorb macronutrients, micronutrients, and even though food is moving through swiftly-ish, right, it does have normal flora. The large intestine, we say abundant microflora because it's quite, I think, I heard it weighs 3 pounds if you took all the normal flora out of your large intestine. So it's going to vary widely between people, but there are anaerobic bacteria there in the genus Clostridium, which we'll talk about Clostridium difficile in this lecture. We'll talk about Clostridium perfringens in another lecture. There's also methanogens, which is part of the archaea. So some people do make methane gas in their intestines. Some fungi present, many, many other species of bacteria. In fact, the gas that we have, right, is called flatus. It's basically produced because we have so many of these bacteria in our gut that will also eat the food that's in there, and they will ferment that food because the colon is going to be largely anaerobic. So think about the oxygen content, right? So we have large oxygen content in the mouth, and as we move through the intestinal tract, the oxygen concentration is going to be lower and lower. So the intestines can harbor these anaerobic bacteria such as Clostridium. And that's why you have a lot of fermentation occur because it's relatively anaerobic. Now, these are some helpful things about our normal flora. We do have lactic acid bacteria that can make basically essential vitamins for us, like B12, folic acid, riboflavin. Those are great. Essential vitamins mean that we cannot make them ourselves, that we need to get these vitamins through the diet, but we have bacteria that can pump those out for us. E. coli also makes vitamin K, which is important for blood coagulation. So there are definitely beneficial bacteria in our gut. And this is our fun fact for the day, that when you go to the bathroom, it's almost 50% microbes in yourself, right? 25, and the rest is going to be your undigested waste. When we talk about foodborne illnesses, there's actually 2 types of foodborne illnesses. There's a infection type and an intoxication type. So the difference is that infections is where the illness comes from the microbe itself. So it's the microbe growing inside of you and causing the infection due to the toxins it's making or just the physical, physically being there. Intoxication type food poisoning is due to a toxin that you ingest. So it's not the bacteria that you eat, it's the toxins that are already in the food. So let's see what's the similarities. The similarities are in the environment. The pathogen contaminates the food. Same thing here, pathogen contaminates the food. The pathogens present at hazardous levels or grows to hazardous levels in the food. So we have a lot of bacteria in the food that we're eating, and then you eat it, right? Here, the pathogen has to grow in the food and then make the toxin. So the load of bacteria is not going to be as much as this, I think, to get the foodborne illness, but it's the toxin production that's important. And the toxin is in the food, and a lot of these toxins are heat stable. So they, if you reheat the food, it does not kill the toxin. And so both of these are eaten. And then for the differences now, you eat the food, this food has a lot of pathogens on it. So some of them will get past your stomach, the acids of your stomach, they're going to get into your intestinal tract. And then they colonize the intestinal tract. So the pathogen is going to grow inside your intestinal tract and can cause irritation, and it can go into the bloodstream. For this one, you have the toxin that enters the bloodstream. So it's not the bacteria itself, it's the toxin that becomes systemic. And then this is also a big difference between the two, is that because this infection, you have to have time for this bacteria to grow inside your intestines. It takes a few days, maybe even weeks for you to come down with symptoms. But since this is an intoxication, it's right away, usually within hours or days. And I know sometimes you're eating at the restaurant or you're eating food and you start to feel sick as you're eating the food. So that's definitely an intoxication type food poisoning. So Staph aureus is going to be our intoxication type food poisoning. So the toxin it makes is called an enterotoxin. All this means is that it's very active inside the intestines. So entero refers to intestines. There's lots of different ones that Staphylococcus makes, and it's not inactivated. So it's very heat stable, like reheating the food is not going to break down the toxin. It's also acid stable, so it's not destroyed by the stomach and it can enter your intestines. This kind of food poisoning is something that we probably all had before, where it's very quick, it lasts a couple days, it's very acute, but it doesn't have any long-lasting issues. So the way you get it, right, is again, food containing the protein is cooked. Bacteria is usually killed. Okay, so when you cook the food, let's say it's chili, the heat is going to kill almost all the bacteria in the food as long as it's cooked to the highest, the high temperature. Containing protein, because protein is going to be a nutrient source for the bacteria. Then you come along with your hands, right? And Staph aureus, as we know, can be a normal flora on our hands, in our nose, in our mouth. So if you're using your bare hands to sort of add cheese or different toppings to the food, you're basically inoculating this almost sterile, right? Because it came from the heat. It's pretty low microbe, right? If any. So then you're putting Staph aureus in here, and Staph aureus has all this nutrition, nutrients to eat and grow. And the important thing about this kind of food poisoning is temperature abuse. So the temperature holding. You really want to be careful with prepared food because they need to either be kept very, very cold or very, very warm. So to either basically on both ends to stop the bacteria from growing. If it's held at a comfortable room temperature, then the bacteria are able to grow and produce toxin. And then the food, then you're eating toxins, and then 1 to 6 hours, you can have that food poisoning symptoms. So, a very sort of notorious thing are like picnics or buffets where you have a lot of food sitting out. They're prepared maybe by your friends and family. No one's really using like gloves or anything. And so this can occur. So here again is our temperature abuse line. No need to memorize temperatures here, but just know that there is a zone between your refrigerator and the holding temperature for foods. This is actually pretty warm, but if you leave it in here for hours, right, you give the bacteria time to grow and also time to produce that toxin. All right, moving on to shigellosis, or otherwise known as bacillary dysentery. So dysentery, this is a bad word, right? This is really severe diarrhea that ulcerates or damages the membrane of the intestine. So you have blood showing up, you might have mucus showing up, so that the lesions are going to, you'll see that in the stool. So for shigellosis, you can have a mild to severe, all these, infections we're talking about, it can move from mild to severe depending on many variables, including your own health, including the strain of bacteria that you have. So many species of Shigella can cause this kind of shigellosis. Shigella dysenteriae, this specific species, is the most severe form of shigellosis, and this is because this strain of bacteria, that species of bacteria, does make something called Shiga toxin. And this toxin, it affects the protein synthesis of your epithelial cells. They basically can't make proteins anymore. It damages them, and then you have this bleeding, hemorrhaging, and lesions in the colon that can result from that. Yeah, so this most severe cases that you can have is that ulceration of the mucosa, dehydration from the diarrhea, and then of course bleeding. An interesting thing about dysentery, and also this is shared with Salmonella and Shigella, that when the bacillus, right, enters into your intestinal tract, it can actually move into the cell. It's taken into a phagosome, and then it kind of wiggles its way out, and then it kind of lives inside your cytoplasm. And then what it can do also is manipulate the protein units in your cells, and it can actually move next door. So it can travel basically from one cell to another cell laterally and never have to leave again. So if it leaves, it's exposed to your immune system, right? So that's not good. So it kind of can travel through cells, just, through the membranes of adjacent cells. The worst thing that can probably happen to you if you get bacillary dysentery is kidney failure, which is called hemolytic uremic syndrome, or HUS. We're going to look at this. There's an E. coli form that also causes this. So the Shigella is it's a reportable illness. However, because you have these milder forms, a lot of people just think it's, they ate something bad, they're sick for a while, it's basically food poisoning, and then they don't, go to the hospital, it's not reported. So there might be more cases than we think. This is a slide on this hemolytic uremic syndrome. So this is, hemolytic means that it's breaking down blood cells. Uremic refers to the kidney. And so the syndrome, right? Syndromes are where you have a collection of signs and symptoms. And so what happens here, this is an example of the E. coli 01157H7. This is going to be the E. coli I was talking about. And, we were also talking about Shigella. So this is another bacteria that causes this. And both of them, right, this HUS is caused by a toxin. So in the Shigella we talked about, it's just called Shiga toxin. And in the E. coli we're going to talk about, it's called virotoxin, but it's exactly the same thing. It's just made from the E. coli. And so here are some signs of the HUS. And we can see that we have breaking down of the red blood cells, which is the hemolytic portion. We have loss of our clotting factors, and we have kidney damage. So this is the, really serious form of infection. And the other bad thing is that HUS, it's mainly supportive treatment. There's nothing really they can do once you have HUS. They just, they'll have dialysis to help your kidneys. Well, because your kidneys are not working, dialysis helps to clean the blood as if it's an external kidney, basically, right? There's no antibiotics for this. So that's why it's so deadly, because if you get it, it's very severe. And there also the, medical community's hands are tied because there's not a good therapy for it. Salmonellosis is caused by salmonella, and salmonella is, so both salmonella and Shigella, right, they're going to be infection type food poisoning because it's a bacteria that makes you sick. So we see a longer incubation time, 12 hours to 2 weeks. And salmonella is found in us, but it's also found in reptile guts, chicken, and eggs. Of course, these are linked. But when the eggs develop inside the chicken, it doesn't develop in a separate area from like their GI tract. So some of the salmonella can get incorporated inside the eggs as they're being created inside the chicken. And that's why salmonella, that's why eating undercooked eggs is somewhat dangerous because of that salmonella. So it's a fecal oral route of transmission because it's in the intestines of these guys. And by the way, if you have a reptile pet, right, if you have snakes, lizard, iguanas, those guys have salmonella in their intestines. So make sure you wash your hands after you handle them. The most severe form of salmonellosis is Salmonella enteritis, very common. The food poisoning, again, from mild to severe. And salmonella is pretty wimpy in terms of temperature. So yes, it can survive in the freezer, but if you cook it at sort of a high temp, it'll kill it. So the salmonella is very sensitive to heat. As long as you cook the foods, it will kill the bacteria. So that's why you want to make sure your chicken is cooked well, your eggs are cooked well. Even though I love sunny side up eggs, there's a risk. There's always a risk. The most serious pathogen of our Salmonella genus is going to be Salmonella typhi. It causes something called typhoid fever. And this strain is only found in humans. And basically asymptomatic chronic carriers do exist. It's very, very serious. The mortality rate is 10% if you let it go untreated. You have a really high fever, body aches, nausea, lethargy, and possible rash. And again, this little picture here looks similar to our Shigella in that this for salmonella comes in and it can enter into these specific cells. They're called M cells. These are cool because in your intestines, you may have learned that there's little areas inside the lining of your small intestine called Peyer's patches, which is where a lot of lymphatic cells are. B cells, T cells, macrophages, all your immune system are like a collection there. And so these M cells are basically a portal into the Peyer's patch. So these M cells are made to grab onto bacteria, to take them in and to pass them on to the Peyer's patch to let your immune system see them. And so these salmonella bacteria, they do enter these M cells, and then from the M cells, they enter the macrophages. And then you can see that from this, they survive inside the macrophages and they can be passed on to other cells. They can go on to infect your lymphatic system. From the lymphatic system, they go on to your blood, liver, spleen. So it can become systemic in this manner. So we can't leave without talking about Typhoid Mary. Typhoid Mary was the first healthy carrier to ever be discovered for Salmonella typhi. And lo and behold, she worked as a cook, which is probably why she was discovered. And over 7 years, from 1900 to 1907, she worked for a number of households as a cook and unknowingly spread illness to the people that live there. And again, remember, this is fecal oral transmission. So, as anyone in this time would think, she's accused of passing on this deadly disease to people she worked for. She herself has 0 symptoms. And so it was very hard for her to believe that she was the one that was causing people to get sick. And she was kind of a recalcitrant, sort of ornery woman. And she didn't really listen to the the police and health official at the time. And so she did have a little short run from health officials. She was captured once and then captured again. When she was recaptured, she was forced to live in relative seclusion until she died. So it's a sad story, but she was the famous first chronic carrier for Salmonella typhi again, and her name is Typhoid Mary. And today we believe that she was responsible for 51 cases of typhoid fever, and then 3 people died from that. All right, moving into our E. coli. So we have seen this guy under the microscope lots of times, tiny, tiny pink rods. And the reservoir is going to be intestines of humans and animals. I should say like warm-blooded animals. The vast majority is fine, right? There's lots and lots of strains of E. coli, and they can be commensal. Some of them might be pathogenic and cause diarrhea diseases, but we're going to talk about the 4 major strains that cause 4 major pathogenic strains that cause disease. So E-tech, E-IEC, E-PEC, and E-HEC. So for the first 2, these are going to be the milder forms. So enterotoxigenic E. coli means that it does make a toxin. All of these guys are fecal oral transmission. So heat stable enterotoxins, right? So that you make a toxin in the food. The toxin does not get killed by high heat. It acts very similar to cholera toxin, which we haven't talked about yet. But what cholera toxin does is it causes your intestines to push out a lot of electrolytes, which would be like sodium, chloride, so salt, let's say. And then once you lose the salt, the water is drawn out by osmosis. So water is forced to leave because it's following the osmotic pressure by the loss of your electrolytes. And so you do lose a lot of fluid in this way. It doesn't damage the intestinal lining, so it's a watery stool, and this is what commonly causes traveler's diarrhea, which is also called Montezuma's revenge. So self-limiting, relatively mild. If you've gone, abroad and you have diarrhea from drinking or eating their food, it's probably ETEC. IEC, enteroinvasive E. coli, is going to invade, right? So we need that invasive. It's going to be similar to Shigella, which causes dysentery, right? So it's going to destroy the intestinal tissue by the intracellular invasion. So again, it goes in, it can spread laterally, it can infect macrophages, travel through the body. So even though this is more invasive and does cause dysentery, it does resolve. So it's usually self-limiting. Watery diarrhea, cramps, malaise, fever, and dysentery can occur. All right, so the next one here is enteropathogenics. The next two are the more serious forms of E. coli infection. So enteropathogenic strain of E. coli is potentially fatal diarrhea, especially in infants and those people who are in less developed countries, meaning that they don't have access to good health care, and they might not have the tools they need to sort of resolve the illness or prevent the illness. So it's very severe kind of food poisoning. One really cool thing that this guy does, and also for EHEC, which I haven't got to yet, is that it really builds like this big pedestal to sit on in your intestines. So there's a protein, TIR, that the E. coli pushes. So this TIR protein is injected into your cells by this bacteria. That protein then turns around and then embeds on the plasma membrane of that same cell. This happens to be what this E. coli attaches to. So it's basically a receptor for E. coli. And on the surface of our E. coli, we have intimin, which is going to attach to this molecule here. So basically, EPEC injected basically its own receptor that attaches to the surface protein intimin on EPEC. And that way we have really tight binding and the bacteria is not going anywhere. So another really cool thing that happens is that there's a chemical reaction that occurs that forces the cell to make all these actin filaments. So this is basically fibers of actin that are pushing down. And what it does is, as it pushes down, it raises the plasma membrane of the cell up. So EPEC is basically sitting on this self-made pedestal or throne, and it just remains firmly attached to cause disease. So let's watch this video.
So here's our bacterium here.
And behind it will be these long strands that are the
They're going to adhere to the microvilli,
these wiener-like structures.
This is what your normal intestinal cell
Comes along here.
Bacterium then causes these things to disappear.
It then starts to nestle down on the surface.
It's locked in.
And then it's going to come down.
And it's going to
set up camp
right there.
We know that happens because we can see this in electron micrographs.
The
microvilli all disappear.
Okay.
And boom. So we're now stuck in the intestine. Bacteria are here.
We're going to zoom in.
Here's our type 3 secretion system. This thing will poke out. We know protein comes out. We know this protein. And then it inserts two more proteins that come out. And down this tube. And they go into insert into the host membrane. So these green proteins will sort of form like proteins will sort of form like a donut core, and this is kind of the tip of the syringe. Then we have to fire in the receptor. Then we have to fire in the receptor. The TEER molecule, and you'll see this red molecule coming down the chutes. Make it then fired into the host cell. Fired into the host cell. Then it goes into the host cell. Then it goes into the host cell. We don't know if it goes through the cytoplasm, goes straight in the membrane. You'll see a little blue dot. You'll see a little blue dot. That's a tyrosine phosphorylation event. Modified. Here it gets modified. Then it ends up in the host membrane. So here we're piling the receptor into the host cell. And then we have to dock TEER with intimate. So we think this tube-like protein retracts. And what will that then do is bring the bacteria down into contact. Here's intimate up here, blue, TEER here, blue, TEER here, contact. We have now landed. Bacteria is stuck on this cell. Bacteria is stuck on this cell. And now we're going to go inside the cell. Now we're going to go inside the cell, and we're going to start to see the face of the TEER that's facing recruits cytoskeletal proteins, and these little donut-shaped things we starting to understand more. Then you're going to see little yellow beads. Then you're going to see little yellow beads. These are actin monomers. These are actin monomers. They then get together and let's like a string of pearls. They polymerize. They polymerize. Flying in, and then start to get longer and longer. Actin does this for living. Actin does this for living. It forms these long projection strings of beads. And what that will start to do is start to put a force on this thing, and you will then start to see the bacteria actually raise up on the cell. And it's because there's actin pushing underneath it that it actually pushes it up into the cell. There it goes. There it goes. And then it starts to push up. Bacteria starts to raise up. Bacteria starts to raise up. It's now building its throne. I'm going to sit up on top of this thing and rule out over it. This doesn't want to make you be a microbiologist. Isn't this a thing of beauty? Isn't this a thing of beauty? And then it just continues to rise and rise and rise. And the end result is we then end up with the bacteria sitting up on this throne, shall we say, to rule out over the mammalian cell. And that's what we see in disease. And that's what we see in disease.
Okay, so that brings us to enterohemorrhagic E. coli, otherwise known as E. coli 0157H7. What do those things mean, right? Those numbers and letters. They're basically markers for surface proteins. Where did I? Oh, yes, here it is. The O is the antigen on the LPS molecule, so it's going to be the carbohydrate part of the LPS, lipopolysaccharide, and the H is going to be the type of flagella protein that it has. So it's type O157 and type H7. This particular strain of E. coli does come in the intestines of cattle. It is not a normal human E. coli flora because it is pathogenic 100%. But it does not affect cattle in the way that it affects our own intestines. Other animals also have it, goats, pigs and deer. So it does make a toxin. We talked about this, right? Virotoxin. It's basically Shiga toxin. And they think that this Shiga toxin, like, this virotoxin, was actually given to this strain of bacteria through transduction, which remember was that when a bacteriophage takes information from one bacteria and can pass it on to another bacteria. In this case, the phage did a transfer of this toxin gene. So, hemorrhagic, right? Entero refers to the intestines, hemorrhagic refers to bleeding, and so we have hemorrhagic diarrhea. Severe cramping, no fever, although it can be self-limiting, but the danger is that it can become very, very dangerous with HUS. It can lead to hemorrhagic colitis, profuse bleeding, and then a large, pretty big number for developing HUS, which we talked about before, the kidney failure. So that's the most sort of virulent strain, extremely virulent strain of E. coli. And it has been in the news a lot. There's outbreaks of E. coli 0157H7 every year. There is a clip. I'm not going to show it now because it's 15 minutes long, but it is from a Frontline. Frontline is also part like a PBS produced show, but it's basically focused on modern meat. This is an hour long if you want to watch the whole thing, but it's basically how we farm our meat today and how it really lends to the creation of and transmission of these very antibiotic resistant bacteria, dangerous bacteria. So I would like you to watch the first 15 minutes. That's linked in Canvas. This is probably before some of you guys were born, but in 1993, there was this huge outbreak of O157H7, and that was at a Jack in the Box. The E. coli outbreak occurred, and then 732 people were infected, and they were all from undercooked beef patties. And they basically skewered Jack in the Box. Jack in the Box had a huge problem with that, obviously. 73 Jack in the Box restaurants, California, Idaho, Washington, Nevada, Louisiana, across the United States, have been affected. And then, because when meat is sourced, right, the Jack in the Box will get their meat from the same area, same plant, and so it can be disseminated to different states. And it's far and away the most infamous food poisoning outbreak in contemporary history. It was huge news back in 1993. And the sad thing is that most of the victims were children under 10, and 4 children died, and then 178 were left with permanent injury, including the kidney, of course, because HUS, and then also brain damage. So it is a very serious strain of E. coli. This is just fun. I just looked up at the CDC. If you look up E. coli outbreaks, you can see, I started in 2006, but you can see that all of this, it's all O157H7. Look at that, right? And it's not just meat, right? There's cheese, there's lettuce. I know a lot of romaine lettuce has, you'll hear about that in the news a lot. Yes, you do have beef patties, but you have spinach, you have nuts. Again, spinach, sprouts, salads, everything. There is ground beef here, but you don't have to just worry about meat or undercooked meat. The E. coli can be passed on to these foods because a lot of times they're, in the packing plant, they're, things can become contaminated. Closer to us, right, we have more E. coli, O157H7. In 2024, we have cheddar cheese, carrots, McDonald's onions, walnuts. So, you have to be careful with everything. Nothing is safe really in terms of food. But you hope that your immune system is good, and you want to wash your vegetables and wash your fruits before you eat them. So let's talk about how these things get contaminated, right? Because the question here is why is a rare steak safer compared to a medium rare or rare hamburger? Okay, so this goes back to the video that you're going to watch. So if you think about the cow, right, the bacteria is in the intestines of the cow. It's not in the muscle, which is what you're eating here. So how does the bacteria get from the intestine and contaminate the muscle? Okay, so this is in the slaughtering process. So when they slaughter the cows, the intestines, you don't do it. I'm guessing, I've never been to a slaughterhouse, but I'm sure things get cut open, the intestines get cut open. And if the cow harbored the O157H7 strain, now you have contamination of the meat. So back to the question of steak versus hamburger. For a steak, this is one muscle from one cow, right? It is intact. This, how many cows do you think went into this hamburger? 1, 10, 100? We don't know. This is a mix of muscle from many cows. And it is the surface area, right? All this ground beef, it has a lot of surface area. So the E. coli can be hiding in the middle of this pile of raw meat. So if you don't cook the middle of the raw meat, you're going to allow the bacteria to survive. So when you have rare hamburgers, that internal temperature does not reach high enough to kill the bacteria, and it can end up eating living bacteria. For this rare steak, the surfaces are cooked, right? So and the surfaces would be the ones that have the contamination of E. coli. So if you cook the surfaces, all the surfaces well, you're going to destroy any bacteria on the surface, and then the bacteria don't really get inside the muscle. It's not a bacteria that squirrels its way inside the muscle tissue. So as long as you cook the outside of the steak, you can have a rare middle, not worry about E. coli O157H7. Medium cooked hamburgers, no, because the browning of the meat occurs at a low temperature, and it's the E. coli can survive that. So you can have brown looking meat, but the E. coli is still alive. Can you get it from eating vegetables? Of course. So this is the thing. How did E. coli from the intestines of a cow get into carrots? A lot of times it's by contamination of water sources. So you might have like a farm here that has your cows, and then you have a waterway, and then there's another farm here that's growing, you know, alfalfa sprouts or some kind of plant. So the E. coli can move through the water, in the contaminated water, and then contaminate the surfaces, right, of the vegetables. And so, yes, you can get contamination of vegetables this way. Can you get E. coli food poisoning from raw milk? Of course you can. So E. coli is in the intestines again. However, cows are messy, right? They don't have a bidet to wash away anything that you know, when they go to the bathroom. So when they go to the bathroom, they're basically standing in their feces, right? So the dirt kicks up, you know, cows walk around, and the dirt will eventually, you know, contaminated soil will eventually like just cover their body. And so when you're milking cows, I'm sure they clean the udders, but you can also imagine that it's possible for E. coli to get from the cow's body into the milk, and raw milk is not pasteurized. Raw milk is just basically cow in the bottle to the store, and it contains a bunch of living organisms. So I recommend highly that you never drink raw milk. It's basically always pasteurize your milk, always get pasteurized milk, because raw milk is very dangerous. Not everybody that drinks raw milk is going to come down with disease, but it's basically like you're playing Russian roulette there. All right, I think we'll stop this and continue with part 3.